My name is Olivia Trapatsos, and my project is the development of a molecular-based detection method for the bacteriocin gene lctA in lactic acid bacteria. The gene lctA is found in some lactic acid bacteria as a bacteriocin, a protein produced by one strain of bacteria to fight off other closely related bacterial species. Bacteriocins are important because they increase the bacterium's chance of survival. The purpose of this research was to create a screening process for lctA in lactic acid bacteria using polymerase chain reaction, or PCR. This research project began as a continuation from a project I did last semester. Last semester, my project focused on cloning lctA from Lactococcus lactis into E. coli using expression vectors. DNA was extracted from Lactococcus lactis, but after many failed attempts to amplify the gene using PCR, it was concluded that the gene lctA was not in this specific strain of bacteria. So the focus of the project then shifted to creating a screening process for the gene in a variety of lactic acid bacteria strains. A local company provided bacteria from a bioethanol fermenter for isolation. Isolated strains of these lactic acid bacteria were chosen for screening. Overnight cultures of the bacteria were initially used for DNA extraction, however, commercial kits failed to yield results. So the cultures directly were used as templates for PCR reactions. Figure 1 demonstrates how the primers bind to the strands of DNA to create copies of the specific region. Figure 2 is the output file from the website used to create the primers. 5 samples were tested for the gene, and none resulted in any products after numerous attempts. Given this outcome, we attempted to determine if we could create degenerate universal primers that would amplify a variety of reported bacteriocins from lactic acid bacteria. The genes GATX and SACR were chosen for analysis along with the lctA gene. The program bioedit was used to display homologies between each gene sequence and then create a consensus sequence. This sequence is shown in Figure 5. Using a consensus sequence would allow for potential conserved regions within these bacteriocin DNA sequences, which would allow us to develop the degenerate universal primer sets for PCR to be done using only one primer set instead of 3 different ones. Based on the result of the sequence alignment, there was not a large enough area of homology to develop this primer set, so separate primer sets for each gene were designed using the website Primer 3 instead of using the consensus sequence. The rest of the semester had planned for testing a variety of lactic acid bacteria strains for the 3 genes, lctA, GATX, and SACR. All 3 primer sets would have been added to each sample during a PCR technique referred to as multiplex PCR, which would enable us to rapidly screen for all 3 bacteriocin genes in one reaction. The results of the multiplex PCR would have been analyzed using gel electrophoresis to identify potential bands representing the target genes. If the gel resulted in a product, further tests would be done to determine which bacteriocin was present in this specific strain of bacteria. I'd like to thank Dr. Andrus for his guidance and support during my research. Thank you.